Have you ever wondered how pharmaceutical companies make sure every tablet, capsule, or injection is safe before it reaches a patient? We ensure quality every step of the way. That one single batch go through dozens of scientific tests involving highly trained analysts, advanced and strict international guidelines before it is ever approved? A tiny mistake in the QC laboratory could affect thousands or even millions of patients. So today, we're going behind the scenes to explore the complete workflow of a pharmaceutical quality control laboratory, step by step. Or someone preparing the entire for a pharmaceutical interview, this video will help you understand the final certificate of analysis. Let's begin.
Every workflow starts with a sample. Raw materials, packaging materials, in-process samples, finished products, purified water, or stability samples, all. But before testing begins, every sample is carefully checked. The analyst verifies
Verifying batch number, product name, manufacturing date, quantity, sample integrity. After verification, each sample receives a unique laboratory number. This number allows come throughout testing. Nothing enters the laboratory without proper documentation. Now the sample is officially entered into the laboratory system. All important information is recorded in laboratory notebooks, logbooks, or
documentation includes sample ID and batch number, date received and required tests, storage conditions, and
not every sample is tested immediately. Some require refrigeration, others need room temperature. Light-sensitive materials use amber containers. Proper storage ensures the sample remains exactly the same as when it was collected. Incorrect storage can completely change the test results.
Before touching any instrument, the analyst reviews the approved testing procedure. These procedures come from pharmacopoeias like USP, BP, EP, or IP.
Standard operating procedures, known as SOPs, are critical. This is method validation. And finally, product specifications. Everything must be ready. We prepare the mobile phase, then dilute it with pure water. Now, the HPLC is ready to run. Before analysis starts, everything must be ready. We prepare the mobile phase and dilute it. Buffer solutions, standard solutions, reference standards, volumetric solutions, and laboratory reagents. All instruments are checked for calibration. This includes HPLC, also GC, UV visible spectrophotometer, and FTIR, dissolution tester, analytical balance, and pH meter. Now comes the most exciting stage, actual laboratory analysis. Depending on the product, different tests are performed. For raw materials, we perform many specific tests. We begin with an identity test. Then, we measure loss on drying. We monitor heavy malleate particle size. We check appearance, weight, and dosage uniformity, friability, testing hardness, content uniformity, and the assay. We begin by checking for related substances. Then, we conduct strict microbial testing. Every result is carefully recorded during testing. No shortcuts are allowed. Testing is finished. But the work isn't over. Now comes one of the most important stages, data review. First, we check the chromatograms for proper separation. Then, verify the spectra for identity. All calculations are accurate. System suitability is confirmed. We must meet the acceptance criteria, check integration parameters, and instrument performance. Unusual results immediately. Accuracy is more important than speed. Now, every test result is compared with the official product specification. Did the assay pass? Were impurities within limits? Did dissolution meet requirements? Did microbial counts comply? If every specification is met, the sample passes. If even one critical test fails, the investigation begins. Sometimes, results fall outside the approved limits. These are called out-of-specification or OO.
The QC laboratory investigates analyst errors, calculation mistakes, instrument problems, reagent quality, and sample preparation.
Environmental conditions, if necessary, testing may be repeated according to approved procedures. Every investigation must be scientifically justified and fully documented. We wear lab coats to maintain environmental conditions. Testing may be repeated according to approved procedures. Every investigation must be scientifically justified, and it must be fully documented. Once testing and review are complete, the laboratory quality control manager or reviewer checks everything again. We begin by reviewing the raw data. We check all calculations, then validate the chromatograms. Full documentation is verified. Every parameter.
Compliance with SOPs and good laboratory practices. Only after complete verification, the results move for final approval.
After approval, the QC laboratory prepares the Certificate of Analysis. This COA document includes specifications, test results, conclusion, and final status. The final decision is simple: pass or fail. This is an official document. Let's look at batch release. If all QC testing meets specifications, the Quality Assurance Department reviews the complete documentation. Only then, the batch is released for distribution. Without QC approval, no pharmaceutical product can legally enter the market. Every medicine you purchase has passed through this rigorous process. QC labs ensure medicines are safe, effective, pure, and consistent. We protect patients every day and compliant with GMP and regulatory requirements. Behind every successful medicine, there is a dedicated team of QC scientists. Precision is our top priority. Every test matters. We follow strict protocols. This is how we ensure integrity. Now you know the complete workflow of a pharmaceutical quality control laboratory, from sample receipt all the way to batch release. If you found this video helpful, don't forget to like, subscribe, and turn on notifications for more pharmaceutical knowledge, QC laboratory training, GMP, HPLC. Thanks for watching, and I'll see you in the next video.